Ladies and gentlemen, dear colleagues and friends, in this video presentation we would like to discuss the role of oxidative stress in the pathogenesis of glaucomatous optic neuropathy. As you know, glaucomatous optic neuropathy has two major components. One is the loss of retinal ganglion cells, their axons, and the other is the tissue remodeling leading to capping of the optic nerve head. And we know today that the entire visual pathway is involved. I would like to explain these different aspects based on the pathogenetic concept of GON we have described a few years ago, which explains both the apoptosis and the tissue remodeling. And as I have said, we would like to emphasize particularly the role of oxidative stress. Why is oxidative stress so important in the pathogenesis of GON? This has to do with the activation of the astrocytes. If astrocytes are activated, they start to produce high amount of nitric oxide, a very small liposoluble molecule with a half-life of about 15 seconds. This small molecule can diffuse in the neighboring axons. Now, if in the neighboring axons the mitochondria produces superoxide, the two molecules fuse and form peroxynitride. Peroxynitride is very damaging because it leads to nitrosylation of functional groups, particularly of SH groups, the so-called S-nitrosylation. Now, as I have mentioned, very crucial is the activation of the astrocytes. We cannot observe that clinically directly in the optic nerve, but we can observe that in the retina. The healthy astrocytes connect the axons with the vessels and form a very regular pattern. If they are activated, they get an irregular pattern, and this is the reason why we see an increased backscatter of light that we can observe in red-free photos on the fundus. But when they are activated, they not only change morphology, they also change gene expressions, and therefore, for example, an increased expression of MMP9. And this contributes to the tissue remodeling and thereby to the capping. But particularly important in this context now for today is the production of nitric oxide. The other question is, why do we have an increased oxidative stress in the mitochondria of the axons? There are several reasons, but the main reason is an unstable oxygen supply. This can, for example, be due to sleep apnea, and we know that sleep apnea is a risk factor for glaucomatous optic neuropathy. But much more important is an unstable ocular blood flow. And unstable ocular blood flow does occur if IOP fluctuates on a level high enough that exceeds from time to time the capacity of autoregulation. Or if blood pressure fluctuates low enough, on a level low enough, again to exceed the capacity of autoregulation. But even more important is the disturbed autoregulation. If the autoregulation itself is disturbed, then each fluctuation of IOP or blood pressure is automatically translated into a fluctuation of ocular blood flow, and this leads to unstable oxygen supply. Now, we have many different reasons for a disturbed autoregulation. In the context of glaucoma, particularly of normal tension glaucoma, the most important cause is the so-called Flammer syndrome. This is the reason why I would like to explain the syndrome very briefly. It is an inborn tendency to react differently, particularly with the blood vessels, to a number of stimuli like coldness or emotional stress. To understand these aspects, I would like to give you first an overview of the cardiovascular aspects of glaucoma. On the one hand, we have the classical risk factors for atherosclerosis, like obesity, hypertension, and so on. And these same risk factors are also risk factors for an increase in IOP, therefore for ocular hypertension, and finally for high tension glaucoma. If you go now to the other end of the spectrum, you have the people who are very slim, low blood pressure, cold hands, etc. These are the people with the risk for a Flammer syndrome, and therefore a disturbed autoregulation. And this disturbed autoregulation finally leads to unstable oxygen supply, and therefore to a chronic oxidative stress in the optic nerve head. And this explains now why the opposition of these different risk factors lead at the end to a very similar type of damage. Now, quickly, some symptoms of FS. They often have cold hands, also feet, low blood pressure, increased pain sensation, silent myocardial ischemia, particularly at night, altered beat-to-beat variation as an indication of the participation of the autonomic nervous system, altered drug sensitivity due to altered expression of ABC transport protein, which also transports drugs. They have a reduced feeling of thirst as a consequence of the activation of the ET1-PGE2 axis. They have increased smell sensitivity, altered sleep behavior, particularly a prolonged sleep onset time. The reason for that is the fact that we all can only fall asleep if our feet reaches a certain temperature. As these people start with cooler feet, they need longer to warm them up, and this prolongs the sleep onset time. They do have a faster pulse width, although the vessels are anatomically normal. This is an indication that they get stiffer. They have a higher spatial irregularity of the retinal vessels, indicating that the dysregulation is not homogeneous along the vessels, and a reduced neurovascular capping we are going to discuss in a moment. They do have more often optic disc hemorrhages, and this is a consequence of a local breakdown of the blood-brain barrier. They do have more often activated astrocytes, and we mentioned in the beginning that this is crucial for the development of GON. They do have also increased retinal venous pressure. On this picture, you see in red the IOP and in blue the retinal venous pressure. At the right-hand side, you see the normals. In these people, the retinal venous pressure is only slightly above the IOP on the average. On the other extreme, on the left-hand side, you see the glaucoma patients with FS. Here, the difference between venous pressure and IOP gets very large, indicating that people who have a dysregulation in the periphery also have a dysregulation of their vessels in the retina, including the veins. And the increased venous pressure decreases perfusion pressure and increases transmural pressure, both contributing to the damage. They have more often retinal vein occlusions. This is due to an increased local concentration of endothelin, which constricts the vein, increases retinal venous pressure, and finally gets in a vicious circle, leading to the clinical picture of a retinal vein occlusion. They do have often, or nearly always, a diffuse component of the visual field defects, visualized with the BBA curve, that fluctuates dramatically over time. They do have a reduced response in the dynamic retinal vessel analysis. On the left-hand side, you see the results of a person without FS, on the right-hand side of a person with FS. Red is the artery and blue is the vein. And you can see, if you flicker light, you get a nice response in FS negative, but a much smaller response in FS positive people. If we flicker light, we get a dilatation of the very small vessels, and then a secondary dilatation of the bigger vessels, as I have shown here. And this is mediated by endothelial cells. And if the response is reduced, it is an indirect sign that the endothelial cells are involved. People with FS have more often an optic nerve compartment syndrome. This means that the cerebrospinal fluid around the optic nerve cannot communicate freely with the rest of the cerebrospinal fluid. This leads, on the one hand, often to an increased pressure, particularly after a Valsalva maneuver, and to altered composition of the liquid. The good thing is that if you treat FS, then most often the compartment syndrome is also reversible. FS occurs more often in females than in males, in sportive than in unathletic subjects, in thin than in obese subjects, in intellectuals than in blue-collar workers. They do have more often and increased oxidative stress, even systemically. This can be indirectly demonstrated by the number of DNA breaks in the circulating lymphocytes with the help of a Comet assay analysis. Now, what is oxidative stress? Oxidation means burning, and oxidative stress is a blaze, is an uncontrolled burning. Oxidation chemically means that an atom like oxygen with a higher electronegativity accepts an electron from another atom with a lower electronegativity, like carbon. And the reduction is the reverse. And this is the basis of the energy cycle. On the one hand, on the right hand, we have the burning in the oven or in our body, in the mitochondria, to produce ATP. In this process, oxygen receives electron and gets bound. In the opposite, we have the photosynthesis in the green leaves. Here the light is absorbed, and the energy then, this light energy is then used to pull back again this additional electron, and oxygen is liberated. Now, why is it exactly oxygen that is so important? Because it is very electronegative and it is readily available in the atmosphere, for example. But then why is not everything oxidized? The reason for that is in the ground state of the oxygen. The oxygen is a B radical, but with a parallel spin. And this leads to the so-called spin restriction. In other words, in the ground state, oxygen cannot easily react with other molecules. We all know that wood burns only after the fire has been ignited. But now, how is oxygen activated? This can be done either by adding energy and turning one spin. This leads to singlet oxygen. Or by adding one individual electron. This leads to superoxide. Both are very reactive and belong to the class of ROS, reactive oxidative species. Now, what is the cause of oxidative stress? There are many, but in the context of glaucoma, the important risk factor is a reversible hypoxia. Mitochondria like to have a constant oxygen tension. If this oxygen tension drops for a while, deep enough and long enough, we get an infarction. In other words, the cell will die. If, however, the drop of oxygen is less intensive or less long, then the cell can survive, but with a burst of free radicals. Why is that the case? This can be explained by the function of the mitochondria. Here you see the inner membrane of the mitochondria, and built in is the respiratory chain. The electrons from the Krebs cycle enter the respiratory chain and travels then from one complex to the other, ending on the oxygen. And the energy is used to pump protons, and the proton gradient drives the ATP to produce ATP. Now, if oxygen drops for a moment, then we get an increased pressure in this chain, and the electrons would like to jump out, but they have no place to jump. If during the reperfusion phase the concentration of oxygen rises again, then individual electrons jump on oxygen and build so-called free oxygen radicals. But Rhos are physiological, yeah, even essential, for example, for infect safe. The critical is the balance. As long as the Rhos production and the coping capacity are in balance, then we do have an ideal situation. If, however, Rhos production increases or the coping capacity decreases, then we get an oxidative stress. Now, this Rhos induce damage to different molecules. For example, in the DNA, they induce DNA breaks. However, we have a fantastic machinery in our body to repair these DNA breaks. They also can damage proteins. These damaged proteins are recognized, then tagged by ubiquitin, and then pulled into the proteasome electrostatically, cut it in pieces, and recycled. So therefore, again, whether we develop a disease or not is a consequence of the balance. As long as the Rhos-induced damage is in balance with our repair capacity, we are in a so-called premorbid oxidative stress. In other words, we do not have a disease. If, however, the Rhos-induced damage increases or the capacity decreases, for example by age, then the oxidative stress causes a disease. Now, this brings us to the restoration of this balance by antioxidants, either by food or by drugs. Now, antioxidants either donate an electron or accept an electron in order to neutralize free radicals. Why can they do that? The reason for that is the fact that polyunsaturated fatty acid, they build so-called electron clouds. The individual electrons are not bound to the individual atom anymore, but build a so-called cloud. This is symbolized in the middle figure. This has two consequences. One is these molecules absorb in a whole range of the light spectrum. I would like to explain this with lycopene in the tomato. Lycopene, the red drawing here, absorbs light in the blue and in the green area, but not in the red area. This gives the very nice color, red color to the tomato. The other property is the fact that these clouds can easier donate or accept electrons. Now you understand why antioxidants are always so nicely colored. An example of an electron donator is epicatechin in the green tea. Or an example of an electron acceptor are the anthocyanin, for example, in blueberries. You know the term polyphenols. This here on the left is a phenol ring. If we have more than one phenol ring, we speak about polyphenols. Again, these are unsaturated and therefore they build electron clouds. And therefore they have an antioxidative property and the nice colors as shown here. An example is the resveratrol in the red wine or the polyphenols in chocolate. There are many more, but now we are coming to the antioxidative drugs. They act principally in the same way. But the molecule needs to reach the right tissue, for example the brain, the right cells, and the right cell organelles. And this is the most crucial point. For example, vitamin C or E do not reach the mitochondria. And they need to neutralize the correct free radical and to be standardized. Ginkgo biloba extract 761 is a standardized extract with qualitative and quantitative constant composition. It contains flavonoids, ginkolits, and bilabolids. In vitro studies have demonstrated that indeed it acts on the level of the mitochondria, it counteracts the oxidative stress and thereby increases the survival rate of the cells. In vivo, in experimental glaucoma studies in animals, it has been shown that the probability for survival of retinal ganglion cells is dramatically increased. In clinical studies, it has been shown that the prognosis of the visual fields in normal tension glaucoma is significantly improved. Ginkgo extract 761 also reduces the systemic oxidative stress, again demonstrated here indirectly by the number of DNA breaks in the circulating lymphocytes with the COMET assay. You see, people under ginkgo have much less DNA breaks. This brings me to the summary. Oxidative stress plays a major role in the pathogenesis of GON. FS reduces autoregulation. This leads to unstable ocular blood flow and therefore increases oxidative stress. EGB 761 is a standardized ginkgo extract reaching the inner membrane of the mitochondria. EGB 761 has been scientifically proven to have an effect in vitro, in experimental studies, and in clinical studies. Thank you very much for your attention. In case you would like to have some more information, we have a book on ocular blood flow and the other on basic science in ophthalmology, and also these two home pages where you find more information. Thank you again.